start streaming now. So after that, I will start. Am I audible to you, sir?
Yes, 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 madam.
Okay, okay, sir. Thank you. Shall we start, Deshmukh sir?
Yes, yes, you start.
Okay. So, a very good afternoon and warm welcome to one and all present over here. On the occasion of 124th birth anniversary of founder president of Shri Shivaji Education Society Amravati, Dr. Panjabra alias Bhausaheb Deshmukh, our Science College Congress Nagar Nagpur in collaboration with Microbiology Society India has started a lecture series which is based on the theme Indians speaking from abroad. So on today's occasion, on behalf of the organizing committees, Honorable Principal Professor Mahendra Dhore Sir of Science College Congress Nagar Nagpur and Honorable President of Microbiologist Society India, respected Dr. A.M. Deshmukh Sir, I welcome all the participants here and the most eminently the guest speaker, Dr. Shivraj Nile Sir, National Coordinator of Microbiology Society India, Honorable Dr. S.D. Patankar Sir, and all the participants who have joined this online meeting. So today's lecture will be on the topic of by Dr. Shivraj Nile Sir, who is Associate Professor of School of Pharmaceutical Sciences, Zhejiang Chinese Medical University, Hangzhou, Zhejiang, China. His topic is Insights on Natural Product Research and Career Opportunities in Life Sciences. So, for the part of the introduction of such an eminent personality, I take this opportunity to introduce our eminent guest speaker, Dr. Shivraj Nile Sir. Shivraj Nile Sir currently working as an associate professor of School of Pharmaceutical Sciences, Zhejiang Chinese Medical University, China. In his academic qualification, he has completed his undergraduate degree from SRTM University, Nanded, Maharashtra. His post-graduation also from the SRTM University, Nanded, Maharashtra. His PhD he has completed again at SRTM University, Nanded, Maharashtra on the topic Studies on Characterization and Inhibitory Potency of Plant-Based Flavonoids and Xanthine Oxidase. His postdoctoral experience is he has worked as Brain Pool Postdoctoral Fellow at Department of Bioresources and Food Science, College of Life and Environmental Sciences, Konkuk University, Seoul, South Korea, from 2012 to 2014. In his working experience, he has served as an assistant professor in SRTM University. After that, he has served as a postdoctoral fellow in South Korea, as well as associate professor in COCS IT College. And after that, now he is working in China. He has supervised many more graduate students as well as PhD students in his career. He has different teaching activities as an biostatistics and basic computer, then bioprocess engineering and technology, basics in biochemistry and bioinstrumentation. He has organized various scientific meetings, as well as he has more than 100 research paper publications in SCI index journals as well as Scopus index journal and many reputed national and international journals to his credit. Apart from this, he has attended and presented many national and international conferences. Yes, he has his books publications also to his credit. Many more books have been published on the topics, different topics of the life sciences, as well as he has an additional research experience. He has worked
You are not audible, Pranita.
Pranita, please unmute. Please unmute, Pranita.
Okay. So,
Okay, madam. No
sir,
problem.
from what time it was, or from the beginning it was
No.
muted?
Just for a second. No problem.
Okay, okay. So he has his additional research experience. He has worked as brain pool postdoctoral fellow at South Korea, as well as he has research experience in the supervision of Professor C.N. Khobragade Department of Biotechnology, SRTM University, Nanded. He is the recipient of many awards and fellowships to his credit. He has received Research Pioneer Award in 2017. He has received Outstanding Research Science Award in 2018, Young Scientist Award in 2017 in an international conference at Korea. He is the recipient of Science Slam Award in 2017 in international conference, Best Oral Paper Award at Aurangabad in 2012. He has received first prize in Best Poster competition in 2012, Best Research Presentation Award in 2011, Young Scientist Award in 2011, again Young Scientist Award in an international conference at China in 2011, as well as he has selected for Young Scientist Award in oral presentation in 2012 in Indian Science Congress. He has received Junior Research Fellowship Award by University Grants Commission, New Delhi in 2007, Senior Research Fellow Award in 2011 by University Grants Commission, New Delhi, as well as awarded by ECLEF Merit Scholarship of Government of Maharashtra, India in 2002 to 2004. He is the recipient of Best Research Paper Award in Bioinformatics. He is the recipient of Best Research Paper Award in Bioinformatics in 2007. He is the board of editorial board member of various journals. To his credit, he has the professional membership of various professional bodies like Indian Science Congress Association, Asian Federation of Biotechnology Korea, Phytochemical Society of Asia, International Society for Research Development, London, UK, the International Natural Product Sciences Task Force. So thank you so much, sir, that you have very great achievements in your career, and we are very much fortunate to have your lecture in our lecture series. And thank you so much for accepting our invitation to deliver your precious talk. So on behalf of organizing committee, I request you to deliver your precious talk, sir.
Thank you, madam.
Before that, it is a kind request that if you can share your journey from India to China, then it will be great for our participants, sir. Thank you, sir.
Thank you, madam. May I audible?
1 minute, sir.
Yes, sir. Yes, sir.
Before you, again, I want to request all participants, please mute yourself. Again, I am giving warning to everyone. Otherwise, I will remove you. Thank you. Thank you, sir.
Thank you, sir. May I audible and PPT is visible?
Yes, sir. Yes, it is visible.
Yeah, first of all, I am very thankful to respected Deshmukh sir. Like, I am the student of Deshmukh sir. And also, I'm very thankful to Professor Dhotre sir, principal of your college. Also, Professor S.B. Patankar sir. And special thanks to Pranita madam for inviting me to present my lecture or my my experience to the students. So I'm very thankful to all the organizing committee members, and also thankful to all students, those are like attending this lecture. So I will start like my presentation. Before I will give brief introduction on my research career. Then I will discuss about my journey from like SRTM University Nanded to Korea to China and other different countries. Also, I will share like different projects or different regarding different fellowships in China, Japan, Korea, all around the world. Then we'll make I will also suggest some collaborative work in between your college and students among the Chinese scientists and Korean scientists. So I will go one by one. First of all, I will discuss my research in brief. Basically, I am working on different areas like natural product, nanotechnology, food biosciences, even along with Professor Arun Kharasar and also from many professors from SRT University Nanded, like Pune University professors. So I am collaborating with different institutes and departments in India, and we are working on natural product and their utilization against microorganisms, different diseases like cancer, diabetes, and gout. So basically, our research focused on natural product and their utilization as a functional food and drug. And also I will discuss the recent career opportunity in life sciences, because this COVID opened a lot of opportunities to life science students and the faculty members, even researchers. So I will discuss all my details one by one. So this is my brief profile, research profile. Currently, we are working on different areas as I discussed with you. So this is my expertise. We are working on extraction of different bioactive compounds from herbs and food products, their biological activity, biological activity evaluation with reference to anti-inflammation, anti-cancer, cytotoxicity, and antimicrobial activity. Also, we are targeting different enzymes because many enzymes are very important in metabolic disorders, like the metabolic enzymes which are responsible for disease development, and also they cause various disorders. So inhibition of these enzymes are very important to cure different diseases and also inhibit the different disorders. Nowadays, many people working on targeted drug delivery system for cancer and gout and various diseases. So we also in our lab synthesizing different nanomaterials to get targeted drug delivery against various diseases. And basically in our lab, we are working on cancer and gout therapy. Recently, we also started working on natural colorants, functional food developments, and metabolic production of phytomedicine. Even we are also working on gut microbiome. Nowadays, it's very important to target gut microflora because that is very important to these gut microflora acts as a marker, markers for disease diagnosis and disease cure, because they produce different kind of metabolites and which can be utilized as a to control different diseases. These are my publication data, record, patents, and also already madam discussed all the things with you. So this is citation index. I got more than 5,175 citation within this tenure of my career. Finished various projects at India, Korea, and even in China. We developed one polyherbal drug therapy for kidney stone, which is very effective and it is already commercialized in India. So this is brief workflow. Basically, we are targeting different herbs and food products and their botanical, physical, chemical, and biological characterization. And we are doing metabolic analysis like extraction and their chromatographic and spectroscopic analysis by using different methods like HPLC, HPTLC, GC, and UV mass and NMR. And we're also doing biometal synthesis to inhibit anti-cancer activity, to check the anti-cancer activity, cytotoxicity, genotoxicity. So these are all biological activity. Before that, we are targeting our drug or extracted compounds in vitro, in silico. In silico, widely used to modify the drug. In vitro, by using different lab techniques, and in vivo by utilizing animal models. So here is the herbal drug development process. Before going for herbal drug against even for as an antimicrobial agent or as an anti-cancer agent, we have to study different characteristics for the standardization and quality evaluation. So we are doing botanical characterization, physical characterization, chemical characterization, and biological and organoleptic. Organoleptic, it's related to food science, that is sensory evaluation. For functional food development, we have to check color, odor, taste, and texture. So it is not much related to current talk. So basically, I will talk on botanical, physical, chemical, and biological characterization. So botanical characterization of particular plant to extract different bioactive compounds, we have to check macroscopic and microscopic evaluation of particular plant, like shape, external size, even qualitative and quantitative analysis, even powder study, because for extraction of bioactive compounds we need the sample in powder form, and the powder quantity is very important. Even qualitative and quantitative method means just we have to screen various bioactive compounds qualitatively and quantitatively. Then moisture content is very important. Extraction value is very important. Then ash value. So these are different physical characters, but the major, main important parameters are chemical and biological characterization. Here in chemical characterization, we are targeting different bioactive compounds through different methods. It is called as metabolic analysis by means of HPTLC, GC, HPLC, and also like NMR and MUS. Then we are doing biological activity. Also, we are checking whether our sample is contaminated with different microorganisms. That is microbial contamination. Then we are doing different toxicological studies, pharmacological studies, also antagonistic studies by using different fungal and bacterial cultures. So this is overall overview about the herbal drug development and its quality evaluation quality evaluation for food and drug development, functional food development. So this is metabolics and biological activities. As we know that here in our lab, we are using food and medicinal plant as a source for extraction of various bioactive compound. So first we have to select plant, and then we have to go for extraction, then extract preparation, and then we have to see we have to do extraction by observing targeted and untargeted compounds. Like in any research, we have to develop first hypothesis, and in that hypothesis, we have to select our targeted compounds, because the plant or the food sample, they contain various kind of bioactive compounds. So targeted compound means which are very important to which are very important to be used as a drug for future studies, also as a functional food. Untargeted means maybe like fiber content, other bioactive compounds like carbohydrates, lipids, and fats. So those are not important as a bioactive compounds to study against various diseases, because those already acts as a nutrition or food compounds. Then we have to go for primary screening. After extraction, we have to prepare the sample. We have to run the HPLC and HPTLC. HPTLC is a form of like a TLC that is not much important in extraction of bioactive compounds. Nowadays, HPLC is very important to initial screening, preparatory phase HPLC and final phase HPLC. Then we are analyzing bioactive compounds depending upon their nature. If there are volatile compounds, we are going for GCMS, and for functional group determination, we are going for LCMS. And finally, structural determination, we are going for NMR, and also IR spectroscopy used to determine the different functional groups. So this is all about the metabolomics and biological activities. And biological activities, mainly we are focusing on antioxidant, anti-cancer, antimicrobial activity, anti-inflammation, and enzyme inhibition. So now I will discuss about our research papers already published in various journals. Basically, we are working on the food products or the plant which are widely used as edible product or edible food in daily daily dishes or daily food. For example, you can see onion, apple, and citrus. We are very familiar that many scientists and researchers, they are working on extraction by bioactive compounds from plants. So it means they are cutting just simply cutting of plants and disturbing biodiversity is not important. So here we focused on bioactive compounds from various waste, because nowadays waste management is very important to control the pollution and all things. So here we targeted. This is one project. In one project we targeted only the bioactive compounds from fruit waste and vegetable waste. You can see we considered onion waste, apple waste, and citrus waste, and we extracted different bioactive compounds from each waste. For example, from onion, we extracted quercetin, monoglucoside, diglucoside, triglucoside. These are all quercetin derivatives. Then from apple pomaceae, we are very familiar that we are just peeling the apple and we are eating the apple inside part. that is edible part, which contains like sugar, but we are throwing the like outer cover. But interestingly, we found that the apple cover, it contains very important bioactive compound that is called as tritryptinic acids, like olenic acid, methylenic acid, butenic acid, and eol. These tritryptinic acids are widely used as a cosmetic agent nowadays to emulsify your skin. So that's why even also it help to regulate your metabolism as a lubricant. We found that apple peel is a very important source of these bioactive compounds. So please eat apple along with peel instead of just peeling and throwing the peel. And also the citrus waste, the citrus waste is widely used in Japan, China, and Korea as a tea. They are not throwing the waste, the peel, the citrus peel. It is called as fluido, pulp, and even seeds, because these like outer cover, the citrus outer cover or the peel, they have various various bacterial compounds like phenolics, but here we targeted carotenoids. As we know that carotenoids are very important factor for our body. So like onion waste, different compounds, pomace, apple pomace, tritryptinic acid, and citrus waste, we selected carotenoids. We did analysis by using HPLC and mass spectra. After like extraction and characterization, we did the different biological activities like anti-cancer activity, enzyme inhibition, and we plotted the enzyme inhibition plot, line or bar plot, and we showed whether these extracted compounds have like a competitive or uncompetitive or whatever the like inhibition pattern they are showing. We have to we showed in our papers. And then finally, we are also calculating IC50, CC50 value of these compounds. So I will discuss each paper in brief overall idea about this research. Combating cancer, it's very important to target the pathways, because the cancer progression depends on the it's a cascade of different protein, or it's a progression of activity of different proteins which involved in progression of cancer. So that's very important to check, upregulation and downregulation of protein is very important. So we have to check whether these proteins involving in cancer progression or 1 minute. So you can see we have to check mitochondrial membrane potential, MMP, because breakdown of this mitochondrial membrane potential is very important to produce reactive oxygen species, which activate the cytochrome C, and which leads to initiation of BCL2 and inhibition of BAX. Actually, BAX and BCL2 are the very important proteins involved in cancer progression. Also, these these protein cascade initiate the apoptosis. I think you all familiar with the apoptosis, that is programmed death cell. So in cancer, 4 phenomena is very important. First, we have to break down the membrane potential of mitochondria. Then second, ROH generation. Third, the different pathways involved in progression of cancer by means of enzymes, then apoptosis, and finally, we have to target cell cycle. This is very important to control, or we have to arrest cell at different phases like G1, G2, M. I think you are familiar with these all phases, like mitotic phases and meiotic phases. So basically, targeting apoptosis and targeting cell cycle ultimately inhibit the cell death. Inhibit the cancer cell, and it causes the cell death. So these are the different metabolites in our lab. We already characterize these metabolites from traditional Chinese medicine, TCM. And as you know that in China, TCM is very popular among the people compared to the Western medicine. Like in India, we are not much interested in Ayurveda, but we have to follow Ayurveda, and we have to utilize these Ayurvedic plants as a for drug development or as a medicine. So we extracted chemtothecin, tensionin, rosmarinic acid. These all 5 drugs we extracted from plants, and we developed one method called as hairy root culture. This hairy root culture is very important because simply if we extract the compound from plant, the concentration of particular drug may be very less. But if we do like a repetition of production of particular drug by using a single explant of plant, that is by culturing in lab, that is called as hairy root culture, and growing different hairy root culture, and then again doing fermentation, and it gives enhanced production of bioactive compounds. So this technique we developed in our lab. So I will explain one simple paper regarding onion, what we did in relation to onion waste. Here we extracted spiroside. This compound is very interesting compound nowadays in our lab, because this compound is water soluble. And as we know that water soluble compounds are very important for drug development. Even the many industries, they want water soluble compound compared to the solvent soluble, like ethanol, methanol, whatever they are. So we extracted this spiroside from red onion. Actually, we are working on different onion waste: red onion, yellow onion, and white onion. So each onion, they have like specific characteristic compounds. So we found spiroside in red onion, and we characterized by means of HPLC. After quantification, we extracted the compounds, and we studied like cytotoxicity at cell dose dependent and time dependent manner. Because in cancer study, dose is very important. Even in microbial study, even in any study, dose is very important, and time, because we have to correlate the effect of drug by comparing time required for drug penetration in particular cell, and second, the dose, that is concentration of particular drug. So, and we studied the apoptosis by means of flow cytometric analysis. You can say this is like late phase and late phase necrosis, and so this color shows, like different color shows whether this extracted compound like spiroside inhibiting your apoptosis at what phase, like necrosis or late phase or early apoptosis. You can see in this figure, our extracted compound inhibiting the apoptosis, initiating the apoptosis in early phase and also in necrosis. This is like early phase, and this is late phase, and this is necrosis. So this is all flow cytometric analysis of like bioactive compound, and we have to we are checking the apoptosis, whether these compounds are like regulating the apoptosis or initiating the apoptosis. Then as I told, we have to check the expression of protein, because already I discussed there are different proteins which are involved in cancer progression, and beta-actin considered as a standard protein, and we have to compare the expression of all this protein. So for that we did Western blotting, and here the Western blotting result shows expression. For example, like Mudeng and BCL2, these two are anti-cancerous, these are anti-cancer protein. So we have to inhibit, because this protein they regulate the cancer progression. And you can see after after 48 hours, there is no any expression of protein. When you compare to control, the Mudeng is there is no any protein band after 48 hours. Same in BCL2, you can see the expression is decreased. We compared all these results with beta-actin, Mudeng, CDK, and we have to increase some protein. So this is all different protein expression pattern for cancer study. And then we also studied the enzymes which are mainly involved in cancer progression, like aromatase enzyme, monoxidase A, monoxidase B, acetylcholinesterase. And we inhibited these enzyme by means of our extracted compound along with extract. We have to check the activity of extracts and compound. That is very important in natural product study. As we know that sometimes the extract, it shows a higher inhibition spectrum compared to the compound. And we compared all our results with standard drugs, the anti-cancer drugs available or the standard enzyme inhibitors available in the market. And in this study, we found spiroside inhibited very significantly all the enzymes compared to the different standards. So this study is already published in Food and Chemical Toxicology. So we can say that the bioactive compound extracted from onion waste is a very promising compound which increase the apoptosis and inhibit like different enzymes involved in cancer development and act as a strong anti-cancer agent. So this is about red onion. Then we extracted bioactive compound. Like in previous study, we discussed about tryptophan acid. In this study, we extracted Q3Z. And the extra study in this, like in this apple pomace, we did cell cycle arrest. In previous like slide, I just showed the like apoptosis and flow cytometric analysis. But here you can see we arrested the cell cycle and we studied all this spectrum by using like flow cytometric analysis. And you can see like G0 and G1 phase in green color, S phase in yellow color, and G2M phase in blue color. And we found that the compound extracted from apple pomace, that is Q3Z. Q3Z treated cells were arrested at S phase and G2M phase. And this phenomenon is very important because, you know, S phase is a synthetic phase. Like there is a synthesis of DNA, the genetic material. And G2M phase is like the cell growing phase. So we have to inhibit these two phases. And our compound, this Q3Z, like arrested S and G2 phase very significantly. And also we studied the flow cytometric analysis, and we found that the Q3Z showed very like promising effect in early apoptosis and late apoptosis. If there is a necrosis, that means our compound is not good. If our compound if our compound shows like apoptosis in early or late phase, that is a very good sign that we can use these natural product as a strongest anti-cancer agent. And this is like dose-dependent and time-dependent activity by using bioactive, like apple bioactive compound. Then, as I told, also citrus fruit, the peel is very important. It contains like, basically we used citrus waste like peel, seeds, and pulp. These are all after like eating eating citrus, we are throwing this waste. Even after like processing for juice, we are throwing this waste. So we utilize this waste and we extracted carotenoids. Basically, cryptoxanthin we extracted from this citrus peel. We used mandarin, the citrus variety, and we extracted by means of TLC and HPLC and LCMS. And after that, we used this extracted beta cryptoxanthin against like different proteins, BAX, BCL2, P53, Cas3, Cas7, and Cas9. These all proteins are involved in cancer cell development and progression. And we found that the extracted beta cryptoxanthin is very effective compound to inhibit the HeLa and MDCK. So here we compared HeLa is a cancerous cell and MDCK is a normal cell. We compared the beta cryptoxanthin effect against both cell line, because we have to check whether extracted compound are like affecting on cancerous cell or also on normal cell. So for that, we are also going for genocytogenetic toxicity for our compound. Then these are like different parameters we used. We did tunnel assay, de-acid ASEAN merged assay to prove whether the beta cryptoxanthin induces HeLa cell and cause DNA damage, which was detected by tunnel assay. And we found that this extracted compound beta cryptoxanthin induces like DNA damage in a particular cell line, that is also inhibit the cell cycle. So overall, it shows that beta cryptoxanthin could be used as a prospective drug for human cervical carcinoma. Also, it generate reactive oxygen species and regulate the BAX and P53 and downregulate the BCL2. Also, it enhances the activity of caspase 3-7. These enzymes are very important to progress the apoptosis. And finally, cleavage of nuclear DNA by means of tunnel assay. So we are doing different step by step. As I told, we are also doing genotoxicity by using alum SIPA root culture. And we are like checking whether extracted compounds affecting on different phases like metaphase and anaphase telophase, prophase. If there is an alteration in like these phases, it means that these compounds are not like good to human body, because this compound, if shows alteration in these phases or chromosomal aberration, that means mitotic index. We are calculating mitotic index with like abnormal phases and normal phases. And we are checking whether these extracted compound, for example, from saffron, altering these different phases, and we are calculating mitotic index. And we found that the extracted compounds from saffron, we also use saffron waste, not saffron, because saffron is very expensive, which is like widely used as a very expensive spice. But during processing of this saffron, there is a huge production of saffron waste. So we extracted bioactive compounds, like promising bioactive compounds, and we studied, excuse me, Hari Krishna. So we studied anti-cancer activity and genotoxicity, and we published this paper in Journal of Food Chemistry. So this is all about routine work like extraction, characterization, and their biological activity. But what about the targeted drug delivery? So for that, we synthesized nanomaterial. Here we used one fungus bacteria compound and one like quercetin extracted from onion. As we know that mycophenolic acid, which like extracted from fungus, is very strong anti-cancer agent, but it has like also strong cytotoxicity. So we have to inhibit the cytotoxicity, and we have to increase the enhanced targeted drug delivery of particular drug. For that, you can see mycophenolic acid and quercetin negative, quercetin positive. Here we synthesize nanomaterial, and we combine mycophenolic acid and quercetin. When like mycophenolic acid without quercetin, it metabolites by cytochrome 450, and it like the metabolism is very high with less viability and less pharmacodynamic action. But if you bind quercetin, the quercetin bind to cytochrome 450 and inhibit the metabolic system of like synthesis of mycophenolic acid in fungus or in cell. So we can utilize these free mycophenolic acid with less metabolism and increased viability. That means it can be used as a used for targeted drug delivery. And we synthesize nanomaterials by using different nanomaterial synthesis method. And we studied drug effect and drug cytotoxicity by using HeLa cell lines. Also, you can see the tumor size. This is like control tumor, and this is free quercetin, effect of quercetin. This is effect of mycophenolic acid. So we like isolated this tumor from rat, and we found that when we combine mycophenolic acid and quercetin with lipopolysaccharide, we combine, and we found the tumor size is very short, very small. It means that the compound conjugated through nanomaterials, or it is also called as combinational therapy, gives very effective efficacy towards the tumor size. Also, there is increased cytotoxicity towards the cancer cell line. It indicates that the combination therapy, that is MPA, LPN, and quercetin, removes the hurdle of individual drug. As I told, mycophenolic acid is a strong anti-cancer agent, but it also has the strong toxicity. So we reduce toxicity by adding the quercetin and by combining these two drugs by nanomaterial lipopolysaccharide. So combination of this this lipid nanoparticle gives higher cancer activity with less tumor decrease in tumor size, and also it is safe for administration compared to this single form of mycophenolic acids. So it means that the nano material is very important nowadays for enhanced drug delivery and targeted drug delivery. Also, we were working on influenza virus, also targeting. We utilize ashwagandha, and we developed one method called as subcritical water extraction for enhanced production of bioactive compounds, and we studied the anti-cancer study. We published one paper in like Impact Factor 17 regarding COVID-19 and the different like cytokinins involved in COVID. And we got more than like 350 citation within this 2 year. So this is all about my research. And as I told, simply cutting of plants or taking explant is not important. So we have to develop some methods for enhanced production, because we are directly connected with industry, because like in China or in Korea, even in India, many industries are interested to make collaboration with different laboratories. And we are developing like enhanced production of active compounds by through hairy root culture. So you can see for hairy root culture, we are using medicinal plant explant only, for example, leaf or any simple part of plant, and we are incubating with Azorobacter bacterium, and we are cultivating this in lab, and we are producing hairy root culture. And after that, we are doing fermentation, and we are extracting bioactive compounds. So this is hairy root culture method for enhanced production of bioactive compound. And after production, we are going for nanomaterial synthesis like liposomes or encapsulation method. There are different methods. And finally, also we are doing biomaterial, means the nanomaterials which can be used as edible sources. For example, we are using gene protein, which was extracted from maize, like corn. And we are binding these different drugs by using this protein. So this is called as biomaterial. And these all like enhanced production of bioactive compound, then nanomaterial synthesis, and then biomaterial synthesis. After that, we are going for different biological activities like anti-inflammation, anti-cancer studies, enzyme inhibitor studies, and we are also dealing with different animal studies. And finally, we are as I told, we are working on spiroside, that is water-soluble compound. So we are also like in China, even in Korea, we are focusing how to how to do commercialization of particular drug. So we are targeting clinical trials for our drug in future. So this is all about my research. So if you have any question, we'll discuss later. Then I will discuss different opportunities. This is like very important part for students. As you know that I am student of SRT University Nanded. I like already Professor A.M. Deshmukh sir. He knows me very well. He's like my teacher. And also I'm familiar with various faculties from Nanded, even Osmanabad, from Aurangabad, even Nagpur, Amravati, Jalgaon. So I am familiar with all universities from our region. And I am also student of SRTM University Nanded. You know that like these universities, we have some like less facility. Even though I did my research in SRT since from graduation to PhD, and I got like one opportunity to do postdoc in Korea, and I utilize that opportunity. So in life science or even in any subject, if you want to do something, you have to wait for one opportunity, and for that you have to do something different compared to others. Then only you can get the opportunity. So there are a lot of like opportunities in life science compared to, you can go to industries, you can develop your career as a research scientist. You can also develop your business, because nowadays the government, actually I heard about some funding. The government is, the current government, they are giving some seed money for development of different businesses. Like one of my friend, he's working in Bangalore. He developed one factory, production of these like mask and lab equipments. So that is also like career opportunity for life science student. So there is huge scope for life science student. So before going for any career in life science, you have to go through all these steps. And already I think you passed all these steps. Like after 12th, some people are going for MBBS, MS, MD courses and all things. And the remaining students, those are not like interested in medicine or engineering, even those not secured position in medicine or engineering, they are coming to BSc or MSc. So after 12th graduation, where just you have to finish BSc, then MSc, MPhil and PhD. After finishing a PhD in any subject, maybe zoology, botany, biochemistry, microbiology, biotechnology, it doesn't matter. because there is a wide scope in science. We are not from like arts, so there is a like huge scope in science. Even in this COVID situation, you can see still the USA, even Japan, China, Korea, they are recruiting various students. The basic lacuna or the big mistake, or maybe the students, they are not aware about the different fellowships in China, Japan, or USA. But you have to fulfill some criteria before going abroad, or even before joining to different national institutes, even ICER, different ICERs, different CSR labs, or different like IITs. So there are like different, even integrated PhD, BTech, and MTech, and all things. So before that, after finishing this, like this is like growth of biologist, science outreach, and science education. Maybe some like this is a tree, how the like one biologist can grow from like both field, in academic field and non-academic field. So after finishing doctorate and postdoctorate, he can go in academic section as a like technician, as a teacher, or in university administration, as a teacher in university, as a researcher, a faculty member. This all different areas. Even like if you compare with non-academic area, you can go as a writer, scientific writer. Even if you after finishing your like biotech or mycology, you can go you can like finish your MBA course in a particular subject, and you can also go in business management, considering which is mainly concerned with life science. Also public health manager, also administrator of different industries, research director. So this is all about non-academic areas. So this is one simple tree which showing huge or wide scope for life science students. Just we have to focus like the area, and we have to decide our aim, what kind of like particular area we want in future. So that is very important. So instead just simply like attending classes and doing practical is not enough. You have to observe daily like development in life science, because currently there are different tools you can use, and the biggest tool is mobile. You can use your mobile for searching different position abroad, searching different position in India, also searching different fellowships in India. So instead of going for use of social media like Facebook, hello. So simply like browsing the different social websites like Facebook, and just making your like wasting your time in entertainment, it's not good. So you can utilize your efforts in searching different kind of jobs or different kind of opportunities abroad. So this is a tree. So basically also you can do different transferable skills like numeracy, practical skills, communication skills, ICT skills, problem solving. So these factors are very important to be like best researcher or to be best scientist. So if you want to develop your like own lab, or if you want to develop as a researcher, you have to go through practical skills, very like skillful practical skill for during your masters, like simply PPTing or analysis of particular sample, or even different microbial studies, antimicrobial activities, even antioxidant or anti-cancer studies. So simple practicals you have to do very carefully, and you have to know each and every step during your practical. So that is very important. Even you have to know like you have to develop your communication skill. You have to be a part of like team with problem solving like factor. You have to be like ICT skill means writing and typing skill. Also you should know that different statistical skill in life science like biostat. And important is teamwork. You have to collaborate with different colleagues, different friends in your team. So this skill is very important to develop yourself as a like strong candidate to get job. So before going to enrolling to particular course, or before getting any opportunity, you have to go through English skill exam. This is very important for many countries like Australia, even Korea and USA. If you want to enroll your name as a master student with fellowship, you have to go through these exams. Basically, IELTS is very important compared to TOEFL and TOEIC. IELTS is widely accepted English skill exam, and this exam contain like academic and general. There are like two exams, academic and general. So basically, like the life science student, they are going through academic, and it is like listening. They have different parts like listening, reading, writing. For academic, listening is 30 minutes. Even for general category, the listening is 30 minutes, and there are 40 questions. So this is all about IELTS exam. And for academic, reading is for 60 minutes, and there are 40 questions. And also writing 60 minutes and 2 tasks, and speaking. So the IELTS is divided into listening, reading, writing, and speaking. And you have to score at least more than 6 band in this exam out of 10. And if you have IELTS score with you, you can apply to various fellowships in Canada, even in Europe, even in Australia. And some countries, they don't need IELTS or TOEFL or TOEIC. Simply, they just want to they want to know whether you are like good in different research like activities. They will ask to write synopsis. So instead of that, they will ask write a small synopsis depending upon your master dissertation or your particular, or you have to explore your idea, because you know every lab in world, they have their own research area. So before joining any lab, you have to know the history of that lab, what they are doing, what is the measure of that lab particularly. And before applying, you have to know your like strong points and weak points, and you have to develop your synopsis according to that lab objective. So that is very important. So this IELTS is totally for 2 hours and 44 minutes plus extra 10 minutes. So this is all about different English skill exams. So you can see that opportunities in life science. Life science is related to industries. Nowadays, the industry acquired life science very like drastically. You can say Bicon also, even Reddy's, Serum, and this COVID. Due to COVID, now the life science student got good opportunities and good exposure in various multinational and national companies. You can like expand your career in biotech industry, microbial industry, or plant and drug sector. Also, drug development, enzyme extraction and purification, vaccine development, cosmetics, and biopharmaceuticals. But for that, you have to know what is exactly mean like biotech or microbial industry or plant sector. And you have to perform all these trainings before joining any lab. Already, there are different programs which was run by DBT, DST in different national laboratories, like 1 month training or 2 months. Even in NCL, they have different training course. Every year they have different courses. I am familiar with that courses. So if you are not familiar with particular instrument, just enroll your name to NCL or any CSR lab. Even you can, like nowadays people just want job very fastly. After like finishing master, they want job without any skills. So that is not good. Even they are demanding a lot of money. But if you have very good skill and if you show something to company, because you know all these professional companies, even the scientific laboratories, they invested a lot of money for product development or process development, and they want a particular person should be with like having soundful knowledge regarding that particular area. So like even in industry, there are they have a lot of money invested on product development, and if you are not familiar with some techniques and development of drug, then that is no use. So that's very important. As I told, COVID-19 expanded much more possibilities for research career and job opportunity for life scientists or life science students. So life science, as I told, basically they can focus on teaching, just qualify NET set and go for teaching. But nowadays we are familiar with the positions available in India. So that's very difficult task, because now like day by day, the number of PhD graduates and number of postdoc graduates are increasing So we have to find scope and we have to develop scope in research, even in pharma sector, food processing industry, bioprocess industry, agriculture sector, or also setting up own project or nanotechnology. For all this area, you need skills. And like if you show something different among the others, then definitely you will get hired by different companies or labs. So that is very important to get opportunity in life science. So like even there are some specified fields, medical, you can work in different medical laboratories, even in veterinary industries, like formulation of different animal food products. So nutraceuticals by using different biological sources. Even biotech industries, research, even in research and development, pharmaceutical industry, you can grab opportunity. Even biomedical engineering, agriculture industry, like development of pesticide, insecticides, and they have a lot of scope. Like in China, like one of Indian guy, just he enrolled as a postdoc and he worked with a formulation of different agriculture product, and now he started his own company in Mumbai. He's from Maharashtra. So such kind of activity is very important. Just we have to like show our interest, or we have to fulfill your dream through your efforts and through your hard working. So that is very important. So these are specified areas like even product manufacturing and production. And so for this, you can grab different funding from different funding sources and governments and all things. So as I told, there are different national, multinational industries, research development, different labs. Also, there are a lot of fellowships given by DBT, DST, UGC. You have to know like different fellowships before like joining to PhD or postdoc. And these are mainly after like in India, you have to crack the NET or say SET or BET, these exams to qualify for PhD or qualify for fellowships like RA position, project assistant positions, and different research grants. And after that, you can enroll your name as a PhD student, then you can do postdoctorate, then scientist, and as a faculty. You can also work as an entrepreneur, or you can develop your own business and like development of biofertilizers and food and drug implement. So you can do like this. So now these are the fellowships which are widely like given by India, China, Korea, and other countries. So I will discuss only 3 countries because I'm familiar with India, China, and Korea, because I worked in India, I worked in China, like currently working in China, I worked in Korea. So if you want to join after your master, if you want to join different like labs, CSR labs, so you have to qualify NET and NET, JRF, and all these exams like ICSR, ICMR, ICAR, DBT, UGC, DST. All these like these are government bodies. They offer different research fellowships. So this is like one of the most important thing for Indian students. Then if you want to come, like if you want to join China, Korea, like don't worry about the national issues. Don't think about national issues, because if you want to develop your career, don't divert towards politics, or do not show any interest in politics. If you want to, like if you are interested in politics, just leave it, and you can devote all your efforts in politics. So I think so I'm saying this because China, they have different postdoctoral fellowships like CASH fellowship, CASH President International Fellowships, even and even they have like huge amount of fellowship for foreigners. Korea also, they have NRF, KRF, KGSP. KGSP is for master student. They can go after BSc, they can go to Korea on this KGSP. I got Brainpool Fellowship. This is for postdoc, like after finishing your PhD, and NRF, KRF, these are like different fellowships after finishing your PhD. Others, like after your PhD, you can apply like searching different like even in Google or in Science Direct, different sources are there. You can just search postdoctoral fellowship, and you can join to different laboratories personally with different professors. And you can also like apply for scientist position in India and abroad, and also faculty positions. And finally, like you can like this is a one website. You can find all kind of fellowships on this website, like Pro Fellows. This website is very important for all the students. Pro Fellows. and you can find all fellowships. This is a dictionary for different fellowships in India, even in abroad. And also you can join my Facebook group. We are like posting different jobs and position here. So basically you can work as like after finishing your PhD or postdoc, you can work as a research associate, RA, or research scientist after PhD, scientist, associate scientific director, project manager, technical service manager, regulatory affairs. So some of these things are related to industry, and some are related to research area, like research laboratories. So like this is all about career opportunities. So before after finishing your master, you have to choose your research topic. That is very important. Like idea or hypothesis development is very important to enroll your name as a researcher. Choose your research topic, then narrow it down and find your niche. Niche means your expertise, or each student have their own expertise, because there are different brains, and different brains have different ideas. So write down all ideas and discuss with your labs, or if you want to apply abroad, you have to make synopsis, and you have to identify the research problem and develop clear-cut research question, create a research design and answer them, and finally write your research proposal and submit to different funding agency, or also forward to different professors in India, China, or in any country. So this is like overall flow chart, how to express yourself to get opportunity abroad or India or any country. So as I told, for microbiology student or microbiological research, food microbiology, medical microbiology, secondary metabolic production from microorganisms, that is also very important area nowadays. And we are, as I told, we are nowadays targeting gut microbiome, we have to study microbial diversity, bioinformatics, biofuels. So this is all related. And these are different positions in industry, like analytical chemist, like atmospheric chemist, biochemist, biotechnologist, clinical biochemist. So food scientist, forensic scientist, material scientist, zoologist, toxicologist, research and development scientist, plant scientist, physician, pharmaceutical chemist, pathologist, nuclear scientist, molecular biologist. So these are different career path for student after finishing master or PhD or postdoc. So I will share all this detail with madam, and madam will circulate among with you. So these are some important industries in India, like AstraZeneca, Bangalore, Origin Limited, Bangalore, Aventis, like Biocon, Bharti. So I think you are familiar with all these industries. So you can see Nicholas Piramal, and I'm very happy that my students already working in various labs. Like in Biocon, you can find at least 10 students from different institutes. Those are already connected with me. Even my classmates, they are working in Biocon. So if you finish your master and if you need help to enroll, to crack such kind of interviews, so I can help. I can recommend your CV. But before that, you have to show something related to that company, some research work or some technology development. Even AstraZeneca in Bangalore, so Bharti Biotech. So these are already various industries which are very familiar in India for production of even serum laboratories, Pioneer Biopharma, Ranbaxy, Sami Labs, Sai Labs in Pune, Nicholas Piramal in Mumbai, Orchid Chemicals in Chennai. So these are different companies. So I don't know whether you are familiar with these companies or different institutes in India. So just read all this detail after finishing your master. So this is all about biotech industry in India overview, how huge market currently available. So this data presented in 2018, the Indian biotechnology industry that was valued near about USD 51 billion in 2018. Now it's maybe more increase. That means 14.7% annual growth. So you can see this is a revenue from different industries like biopharmaceutical, bioagriculture, bioservices, and bioindustrial. So it indicates that the life science student can apply or can enroll his name in any area and he can grab opportunity in any area. So this shows how the Indian biotechnology sector is developing. This is only for Indian biotechnology sector. You can, if you visit different countries, or if you want to plan abroad, your job abroad, you can get like huge scope in USA, in Australia, because in China, Japan, Korea, Australia, USA, I observed one thing. Here, the students, after finishing their masters, they are getting job, and they are not interested to enroll their name as a PhD student. They are not at all interested to do PhD. So that's why if you are not happy, or if you are unhappy with your net score, or by chance if you fail to get net and said, just apply for different fellowships in abroad. And many labs, they want Indian students because they know Indian students are very hardworking and very prompt, and they are very knowledgeful, theoretical knowledge they have. So that's very good, good opportunity for Indian students. So even many businessmen, even many governments, they are investing a lot of money in biotech market. You can see where and how investment opportunities like clinical trials and drug development in R&D sector, biosimilars in biotech parks, means vaccine development and other even antibody production. Currently, my wife, she is working in Korea, and she is working on antibody production for COVID-19. So in future, if you want to work on such kind of area like antibody production or some nutraceutical enhancement, immunoenhancers, so that have huge scope. You can see internal biotech firms, 100% incorporated entity, wholly owned subsidiaries. So these are different factors which is very important for limited financial risk and legal risk. You can also collaborate with different foreign companies. So there is a huge scope in life science. You can see I started my journey from Nanded. Now I am the one of the best leading scientists in China also. Just I have showed my picture, and I'm working as a editor for various journal. And recently, I got different projects and recognition from different China, Korea, and India. So I say, if you follow proper guidance, for example, I'm very happy with the A.M. Deshmukh sir. He is doing tremendous job in India. He spreading life science and microbiology around the India through seminars, and that's very important, seminar, presentations and all things, because through seminar you can connect with people, you can get collaborated with people. Through seminar you can get new knowledge or new ideas. So that's very important, and I'm very thankful to Deshmukh sir and all the members of Microbial Society of India. And I'm very happy that I am a part of such a team, and I'm always here to help students from Maharashtra, students from India, to grab opportunity. Even I already recommended many students in Thailand also. Still, Thailand need many students. If somebody is interested, they can contact with me. Even Korea, many professors, they need students. But do not follow different, there are different agents. They are just taking money from students, and they are just saying we are enrolling. So do not follow such kind of agents. Simply search in Google different professors or labs, and you can apply, because if particular professor they have funding, they definitely they will process your application. So, and if you have any problem, discuss with me or even madam with A.M. Deshmukh sir. They are already have tremendous knowledge about life science and all things. So this is all about my research and job opportunities. Now, if you have any question, we'll discuss if possible. Thank you. Thank you all.
Yes, sir. Thank you so much, sir, for your thorough information which you have shared with our participants on the topic of insights on natural product research, in which you have given detailed information regarding anti-cancer drugs and targeted drug delivery system and various aspects of that particular research which you have done. You have a great knowledge which you have shared with our participants. It would be really helping for our student participant, as well as you have given much more idea about career opportunities, not only in China, but in Korea, as well as Japan and USA too. And you have guided our student participants about various opportunities, how they can grab, what should be the steps undertaken by the students, and the various websites and links which you have provided. So thank you very much, sir, that your lecture would be the great opportunity for our students also to pursue their career in abroad. So this is a kind request to you, sir. If you could share your email ID in the chat box so that every participant can be accessible for it in future also. And the participants are requested to type their question in the chat box so that we can ask the queries to our eminent speaker.
1 minute, Pranita.
Yes, yes, sir.
Yes, so today I have heard complete lecture and I was highly impressed regarding your research work, and you have told about career opportunities in biotechnology. Really, it was a very, very informative,
Thank you, sir.
and it depicts your personality, because in what way you have developed in Korea, and today you are having a good position in China. Really, I will say it is a flourished branch of India
Yes,
growing in China.
yes, sir.
So you have also told that there is a tremendous scope in biology, and particularly after COVID-19.
Yes,
So our Indian students must exploit, and researchers like you are there to guide them. So I'm really very happy. We'll remain connected in the future also. And I wish you all the best.
thank you, sir.
Yes, for your future in China, and you will do more and more research in the China.
Thank you, sir. Thank you, sir.
Thank you. Thank you.
Thank you, sir. Thank you.
Yes. Now the topic is open for discussion.
Sir, it's a very nice and informative talk given by you. And
Thank you, sir.
actually, students must be enlightened on the allied subjects and then the opportunities which are present in all the life sciences, and there should not be any such type of the thing that microbiology should go for microbiology and zoology should go for zoology.
Yes,
Our students, they are making a mistake. I make this statement with a very confidence, because I come across the students. They ask always, sir, I did microbiology, I did this, I'm not getting opportunity. But I always tell them, go for such allied subjects and then the different branches. You develop your skills and other things. This is what actually it is needed.
Sir, actually, I did my graduation in microbiology, botany, and chemistry. Then, I did MSc biotechnology, but I then moved to food science. Then I moved to nanotechnology, and now I'm working with COVID-19 and virology.
Actually, in the food science and in the phytochemicals, and there are so many opportunities are there. One thing I want to share, discuss with you or share with you, that is much less work it is going on, or it has not been done in the Ayurvedic preparations.
Yes, sir. Yes.
Yeah, and one thing it is there, the Ayurveda also includes certain type of the fermentation preparations, that is Asava, Arishta preparations, and much less knowledge it is there, and how the biotransformations are going on in this particular type of the processes, that is not known. So it happened like that when you try to identify the certain type of the transform phytochemicals, because one phytochemical you find at one particular stage and then disappears and some new comes. Yes. But unfortunately, in the many laboratories, the HPLC library, the reference library doesn't contain those phytochemicals and they remain unknown.
No.
So they are still undetected and it is required. I just request you if you can do in this.
Yeah, definitely. Sir.
Yeah.
Yeah, definitely.
Because I worked on this particular area.
Yes, sir. Actually,
yeah.
you know, sir, in China, these people are like, for example, if somebody is visiting to particular hospital, they will ask TCM, traditional Chinese medicine.
Yeah.
And even the doctor, they prescribe TCM.
TCM.
So 70%, it's like Ayurveda, Chinese Ayurveda.
Yeah.
But you know, as like many Indians, they don't believe on Ayurveda.
Yeah, never be.
But the Ayurveda is like main root of our medicine.
Exactly.
And that's very important. You can see the Ramdev Baba.
Yeah.
Like I'm not like doing any conflict, but you know, I don't know he's having any knowledge, scientific knowledge about the drugs, but his industry nowadays it's like booming.
Yeah,
around 500 crore per year turnover. So we can think like we are experts in particular area. Like,
there are certain companies or there are certain manufacturers who are working for 100 years, but they are not so much famous.
Yes,
I know, and I have a connection with all such type of the Vaidya, particularly they are called in a proper way, and they are doing their practices, but unfortunately they remain untapped.
Yes, sir. And actually my motto is we have to explore the knowledge from these Vaidya people.
Yeah,
because you know this is a hidden knowledge, and you know it's like just specific to particular person, a particular Vaidya. So we have to like explore that knowledge from Vaidya, and we have to make one platform, a scientific platform to explore, like extract this knowledge, and we have to develop some drugs. And I'm doing in connection with like hair loss and fertility. So in future, but let's see what will happen. Thank you, sir.
Dr. Nile.
Sir.
Sagarika is asking some question. Sagarika, please. Sagarika, ma'am, you can ask the question.
Yes, madam.
She has written in the chat box.
Actually, yes, she has written in the chat box that she is a DST woman scientist at ICMR NIRT. She would like to connect with our guest speaker to discuss her research proposal in which she has to pursue for translational research.
Hi, this is Sagarika. Hello.
Hello, madam.
Yeah, thank you very much, sir. It was a very informative session, and I really thank the organizers who have been continuously enlightening us with all these opportunities. I am a woman scientist with DST, and I'm working with National Research Center in Chennai for tuberculosis.
Okay.
So my current proposal, actually, I have isolated a molecule which is water soluble and highly active with anti-tuberculosis activity, and I really wish to take it forward. And I'm desperately looking for some good opportunities where I can connect. So in this regard, I would really request if we could collaborate or maybe discuss the proposal sometime. If you could share.
Yes, madam, definitely. Just send me the like brief synopsis or brief like work idea. So I will discuss with professors from even USA or India, or like we'll connect with different laboratories, because you know nowadays it's very important to collaborate with different people and different area like nanotechnology, phytochemistry for drug development. So that one is good idea, madam, and I'm happy that you are doing very nice work, and I will be there to help you in this regard.
Thank you very much. If you could please send us your email in the chat box, then I would get in touch.
Yes, madam. I will see.
Thank you so much, sir. Thank you.
Then
yes,
Vivek
sir,
Upasani. Hello, Vivek Upasani. Then Rathisa and Shukla, Dr. Shukla, all they have responded positively and appreciated your lecture.
Thank you,
yes,
sir.
And one more scientist from Nigeria, Dr. Ajay, he is here today.
Yes, sir. Yes, I saw him.
And he also want to connect you in future for academic collaboration.
Yes, sir. Anytime, sir.
I will give him your email ID for further contact.
Thank you, sir. Already I shared my email, both emails. In chat.
Okay, very good, very good. Yes.
sir. Really,
Pranita-
there. Yes,
Pranita
sir.
carry. Ah.
So there are much more messages that the lecture was too good and very much informative, sir. And it would be very much helpful to grab the opportunities to our participants. So very much thankful on behalf of the organizing committee of Science College, Congress Nagar, Nagpur's Honorable Principal Professor Mahendra Dhore, sir, and Microbiologist Society India Honorable President Professor Arvind Deshmukh, sir. So on the behalf of the organizing committee, I am very much thankful, Dr. Shivraj Nile, sir, for your precious lecture and for guiding our participants. Thank you, sir.
Thank you, madam. Thank you, sir. Thank you. Bye-bye.